All right, let's talk about a bug that's a fantastic case study in the world of clinical microbiology. Salmonella enterica, Serovar Manhattan. If you're working in this field, you know how critical it is to connect the dots, from that first suspicious colony on a plate, all the way to a full-blown public health alert. In this explainer, we're going to walk that exact path. We'll start with its basic profile and follow it all the way to its role in major outbreaks. So let's get into it.
And just so you know where all of this is coming from, everything we're covering today is based on a comprehensive Gemini deep search. For any of you who want to really geek out and go deeper into the primary literature, you can find a full list of all the sources in the notebook document that goes along with this.
So here's our roadmap. We're going to follow the journey of this organism, starting right where we're most comfortable, at the lab bench, looking at its basic profile. From there, we'll head over to the bedside to understand the clinical side of things, talk about treatment and resistance, and then finally, we'll zoom all the way out to look at its impact on public health.
Okay, first up, the Manhattan profile. Let's get back to basics. Before we can really appreciate how it causes problems out in the world, we have to know exactly who we're dealing with in the lab.
So who is this character? Well, pop it onto the scope and you're going to see your classic gram-negative rod. It's a zippy little thing too, highly motile thanks to all its flagella. But what really makes it so tough is that it's a facultative anaerobe. It couldn't care less if there's oxygen or not, which makes it perfectly designed to survive the journey from some contaminated food all the way into the human gut.
Now let's talk plates. How do we spot it? On mac and key agar, it's going to be your typical non-lactose fermenter. Pale colonies, nothing too exciting. But where it really starts to show its cards is on selective media, like XLD or hecto and enteric agar. These plates are designed to detect hydrogen sulfide, and Salmonella enterica is a producer. And boom, you get those beautiful, classic black-centered colonies. That's our money shot, right? That's the signal that makes you start thinking Salmonella.
To really nail the ID, of course we have to look at its biochemical fingerprint. Your TSI slant is going to give you that textbook alkaline over acid reaction with gas and H2S production. The IM-Vig tests help us separate it from its other enteric cousins, and it's urease negative. Now, an important point here is its intrinsic resistance. Remember, as a gram-negative, its outer membrane is like a brick wall for a big molecule like vancomycin. So seeing vancomycin resistance isn't some scary new trait. It's just basic microbiology. It's supposed to be there.
So that's the classic way of doing things. Solid, reliable. But technology has kind of changed the game, hasn't it? Let's look at how lab detection has evolved, taking us from plates to pure genomics.
For what feels like forever, culture was the absolute gold standard. It's slow, sure, but you get that beautiful, tangible isolate, which you absolutely need for susceptibility testing and for public health surveillance. Then along come CITs, these culture-independent tests. They're ridiculously fast, giving you an answer in hours. But here's the catch, and it's a big one. You don't get an isolate. That's why you hear public health folks screaming from the rooftops about reflex culture. We still need that bug on a plate.
And this really leads us to how we play detective during outbreaks. Many of us grew up with pulsed field gel electrophoresis, or PFGE. It was like getting a blurry fingerprint of the suspect. But the absolute game changer has been whole genome sequencing. WGS isn't a fingerprint, it's the entire genetic blueprint. With that data, we can get the serotype, predict resistance, and map out relationships between isolates with a precision that PFGE couldn't even dream of. It's just no contest.
Okay, we've spent enough time in the lab. Let's shift our focus to what this all means for the patient. Let's move to the bedside and talk about the clinical picture.
For most healthy people, this is going to be a miserable few days of classic gastroenteritis. It's acute, it's self-limiting, and then it's over. But for the very young, the very old, or the immunocompromised, this bug can be a whole different beast. It can go invasive, get into the bloodstream, and set up shop in other parts of the body. And even when you think you're in the clear, it can leave behind a nasty little parting gift for some people: post-infectious reactive arthritis.
So, what's its battle plan? How does it actually make us sick? Well, after you ingest it, it has to survive the acid bath of the stomach. Then, in the intestine, it deploys this incredible weapon called a type 3 secretion system. Think of it like a molecular syringe. It literally injects proteins into our own cells, tricking them into swallowing the bacteria. Once inside, it hides out and replicates, and this invasion triggers an all-out, massive inflammatory response from our immune system. In fact, it's mostly our own body's shock and awe response that causes the symptoms we feel.
Which naturally leads us to the million-dollar question: how do we treat it? And the answer, as it so often is,
Well, it's complicated. It really depends on the patient and on local resistance patterns.
Now, this is maybe the single most important takeaway for treatment. For your average healthy person with straightforward gastroenteritis, you do not reach for the antibiotics. The cornerstone of therapy is supportive care, basically just rehydration. Using antibiotics won't make them better faster, and you might actually make things worse by prolonging how long they shed the bacteria.
But when things do get serious, that's when the guidelines kick in. And they're not all the same. Take the Dutch guidelines, for example. In a big 2023 update, they changed their recommendation for hospitalized patients. Why? Because ciprofloxacin resistance is becoming a real problem, so they've pivoted to third-generation cephalosporins. And for the pediatricians, the rule from the American Academy of Pediatrics is simple and strict. Any baby under 3 months old gets treated, usually with ceftriaxone. The risk of invasive disease is just too high to wait.
All this talk about resistance circles right back to us in the lab and our role in guiding therapy. And this is where the breakpoints from EUCAST, the European Committee, get really interesting and show some savvy clinical thinking.
All right, pop quiz for my fellow micro nerds out there. You're looking at your EUCAST table for Enterobacterialis. Why is this ciprofloxacin breakpoint for Salmonella species so ridiculously low compared to the others?
And the answer is all about clinical outcomes. The data is crystal clear. For Salmonella infections, even a tiny little creep upwards in the MIC for ciprofloxacin is a huge red flag for treatment failure. So EUCAST set the breakpoint super low on purpose. It basically acts as a tripwire. The second an isolate shows even a hint of reduced susceptibility, we call it resistant and tell the clinicians they cannot rely on that drug for a serious infection. It's a smart, protective strategy.
Okay, for our last section, let's pull the camera way back. Let's move beyond the single patient and the single lab result and look at the bigger picture, public health and prevention. And this quote just nails it. Think about it. Salmonella Manhattan isn't one of your top 10 everyday Serovars. There isn't a lot of background noise, so when a few cases suddenly pop up, it's not just random static. It's like a bright red flare going off in the dark for public health. It screams common source outbreak. And we've seen this play out in the real world. Past outbreaks were tracked using the older PFGE method, and it did an okay job. But here's a cool story. Investigators went back and reanalyzed an old Italian outbreak using modern whole genome sequencing. And it turns out PFGE had mistakenly lumped in an innocent bystander, a case that just happened to have the same fingerprint but was totally unrelated. WGS cracked the case, cleanly separating the true cluster. It's a beautiful example of the power of this technology. So, how do we stop this thing from happening in the first place? It all comes back to the basics. For everyone at home, it's that classic food safety mantra. Clean, separate, cook, and chill. Simple, but it works. In a healthcare setting, we use standard precautions for everyone. But if you have a patient with salmonella who is incontinent, you absolutely need to add contact precautions, gloves and a gown, to stop it from spreading. And this all leaves us with a really fascinating and kind of challenging question for the future. With whole genome sequencing, we can now definitively link two cases on opposite sides of the country, months apart, to the exact same contaminated source. Our old definitions don't really fit that scenario anymore. So the question I'll leave you with is, what even is an outbreak in the genomic era? And there you have it, our tour of Salmonella Manhattan, from the culture plate to the population. I really hope you found this useful. If you did, do us a favor and hit that subscribe button for more deep dives like this one. And please, drop a comment below. I would love to hear your war stories or experiences with this particular bug. Thanks for tuning in.